The overall goal of the following experiment is to assess the level of co-localization of internalized bacteria Neisseria meningitidis with the cytoskeletal protein α-actinin. This is achieved by infecting cultured human brain microvascular cells with an overnight culture of bacteria for 3 to 8 hours to allow the bacteria to enter the target cells. As a second step, cells with intracellular bacteria are washed, fixed, and then permeabilized, which prepare the infected culture for immunostaining of internalized bacteria and intracellular α-actinin. Following the confocal imaging and application of co-localization software, images of the intracellular co-localization can be observed and quantified. Results are obtained that show meningococci expressing OPC and outer membrane protein can specifically interact with intracellular α-actinin based on confocal microscopic visualization and by quantitative co-localization analysis.
Hi, I'm Isabel Murillo from Professor Mamta's Birch's lab at the Department of Cellular and Molecular Medicine at the University of Bristol. Today we will show you a procedure for estimation of the level of colocalization of intracellular bacteria with cytoskeletal proteins. We use this procedure in our laboratory to study the interaction of Neisseria meningitidis expressing an outer membrane protein, OPC, with the cytoskeletal protein α-actinin. So, let's get started.
This work needs to be done in an adequate safety laboratory. 2 days prior to the immunostaining procedure, inoculate the bacterial stain of interest on brain-heart infusion agar plates supplemented with 10% heated horse blood. Grow overnight at 37 degrees Celsius in a 5% carbon dioxide atmosphere. On the day before immunostaining, use a 10 microliter culture loop to make a suspension of the overnight bacterial culture in 2 milliliters of PBSB. Leave the suspension to stand for 5 minutes at room temperature to allow large bacterial aggregates to settle. Next, transfer the top 1 milliliter of the suspension into a sterile tube without disturbing the pellet. Solubilize bacteria by adding 1 milliliter of 1% STS 0.1 molar sodium hydroxide to 2 vials containing 20 microliters of bacterial suspension and invert the tube to mix. To estimate bacterial numbers, measure the nucleic acid content by determining the absorbance of the solution at 260 nanometers. Adjust the bacterial suspension to the required density in infection medium for infecting the human brain microvascular endothelial cells. Human brain microvascular endothelial cells, or HBMEC, are seeded 2 days prior to immunostaining. Place 16mm glass cover slips into the wells of a 12-well plate and seed HBMECs at 50% confluence on the cover slips. Incubate overnight at 37 degrees Celsius in 5% carbon dioxide atmosphere. On the following day, infect cells with the freshly prepared bacterial suspension. In our laboratory, an infection ratio of 200 to 300 colony-forming units per target cell is routinely used. Incubate for 3 to 8 hours at 37 degrees Celsius in 5% carbon dioxide. At the end of the infection period, wash cells 3 times with PBS and fix in 500 microliters of 2% paraformaldehyde for 30 to 45 minutes at room temperature. After washing off the paraformaldehyde, permeabilize the paraformaldehyde fixed cells by incubating in 0.1% Triton X100 diluted in PBS for 10 minutes. Finally, wash the samples 3 times with PBS and proceed to the immunostaining, as shown next. Staining of intracellular bacteria and α-actinin can be performed simultaneously in 12-well plates. Block the cover slips containing the permeabilized cells with 500 microliters of 3% BSA PBST for 30 to 60 minutes at room temperature. After washing with PBS, transfer each cover slip to a new dry well in the 12-well plate. Add the primary antibodies against bacteria and α-actinin simultaneously. 80 to 100 microliters of antibody per cover slip is sufficient if added carefully to cover the surface of the cover slip. Incubate for 1 hour at room temperature. At the end of the incubation, add 200 microliters of PBS to the well. Lift the cover slip and place it in a new well containing 500 microliters of PBS. After 5 minutes, remove the PBS by pipetting and then add fresh PBS. Repeat twice and transfer cover slips to a new dry well. Next, add the appropriate secondary antibodies conjugated to different fluorochromes diluted in 1% BSA PBST. Incubate at room temperature for 1 hour in the dark. At the end of the incubation, wash as shown previously. Then, counter-stain DNA with DAPI for 5 minutes at room temperature in the dark. At the end of this incubation, wash the cover slips in PBS once and then mount with a drop of mounting medium. Mounted cover slips should be stored in the dark until they are ready for observation under the microscope. We observe and capture images of our immunolabeled samples using a confocal laser scanning microscope attached to an inverted epifluorescence microscope. To begin the CLSM procedure, add a drop of immersion oil to the objective and place the specimen slide, cover slip down, on the microscope stage. Set the microscope to a visual mode and find the area of interest using the eyepieces of the microscope. We are now ready to use the Leica software. Select the XYZ acquisition mode. Select the 512 by 512 format. For co-localization studies, the higher the resolution, the more accurate the image. Then select the bidirectional X mode, which will increase the scanning speed and help reduce photo bleaching. Set up sequential scanning settings. Click in the Sec function and then select Between Lines. Select laser beams according to the fluorochromes conjugated to the secondary antibodies. 405 nanometers for DAPI, 488 nanometers for Alexafluor 488, and 561 nanometers for TRITC. Activate photomultiplier tube 1, 2, and 3 respectively. Set up top and bottom of Z-stack source series. Next, set Z-step size to 0.2 micrometers. Neisseria meningitidis is a diplococcus, and since each coccus has an approximate diameter of 0.5 micrometers, a Z-step size of 0.2 micrometers enhances the probability of scanning each coccus at least twice. Set the final scan parameters by selecting line averaging of 3 to improve the signal-to-noise ratio. By clicking Start, double or triple stained images are obtained by sequential scanning at different wavelengths, and by using the Between Lines mode for each channel to eliminate crosstalk between the different chromophores. For an indication of co-localization of two fluorochromes, select the Overlay function to merge selected channels into a single image. For example, when both Alexa 488 and TRITC fluorochromes co-localize, yellow color will appear in the overlaid image. For a 2D reconstruction required for visualizing possible co-localization, compile Z-stack source series using the maximum projection function. After acquiring the Z-stack source series, process your data to obtain an orthogonal image for visualization of intracellular localization of various elements. Quantitation of co-localization is performed using Volocity software from Imprevision. To begin, create a library with CLSM images. Select Extended Focus from the image in the top bar. This tool will combine Z-stacks in a 2D image to be analyzed. Now select the Co-localization tool. The two channels to be analyzed should have the same color depth. Select the area that is going to be quantified. Set the threshold to remove any background. Create the co-localization output by selecting Generate Co-localization. Co-localization statistics will be generated for the regions of interest selected previously. Next, select Mander's coefficients R and My. Finally, export all statistics values to an Excel document for data presentation. Shown here are representative confocal imaging results of human brain endothelial cells infected with meningococci for 8 hours. The location of bacteria is shown in red, while the location of α-actinin is shown in green. Arrows indicate regions where a high degree of α-actinin accumulation appears to have occurred around bacteria. In this overlay image, there are several regions in which yellow-orange colour appears, suggesting co-localization between meningococci and α-actinin. In this next image, optical dissection of an infected HbMEC monolayer indicates co-localization around intracellular bacteria located at the base of a cell. When three-dimensional images of infected HbMEC monolayers were processed, an oblique view of the apical surface shows adherent bacteria stained red, indicated by the red arrow, whereas several bacteria located towards the basal surfaces of endothelial cells, indicated by the yellow arrow, are distinctly orange-yellow in colour. Basal location can be more clearly seen in this end-on XZ cross-section. In contrast to α-actinin, experiments in which labelling of internalized bacteria and either vimentin or actin was performed reveal occasional co-localization with actin, but rare co-localization with vimentin in HbMEC cells. As observed in the previous experiment, α-actinin was concentrated around several internalized bacteria, indicated by the white arrows. The data were also analysed to obtain relative overlap coefficient R, which according to Manders represents the true degree of co-localization, i.e. the number of pixels that co-localize compared with the total number of pixels, and MY values for α-actinin, vimentin and actin. Overall, in HbMEC infected with OPC expressing meningococci, greater than 25% overlap of α-actinin and meningococci was obtained. The coefficient MY is a measure of the frequency of occurrence of the more abundant α-actinin signal each time the less abundant meningococcal signal occurs. This measure shows a striking level of occurrence of α-actinin in the vicinity of internalized meningococci.
We just show you how to visualize and quantify interaction of internalized bacteria with cytoskeleton elements. When doing this experiment, it's important to remember to follow the fixing and immunostaining protocols strictly to maintain the integrity of the structures and to avoid a high background. So that's it. Thank you for watching and good luck with your experiments.